Hello friend, this is AIIMS classes that deals with the entrance exam related to NEET, AIIMS, NISER and Olympiad given by the teacher by AKB sir. You can visit our site and get all information regarding your MCQ questions, your online classes, or you can take study materials directly from the site. Now let's start with the chapter that is called as Mendelian genetics, which is very interesting and very contrasting chapter. Most of the questions normally asked in your entrance exam. So this Mendelian genetics, before going to this Mendelian genetics, first we should know what do you mean by genetics? What is the meaning of genetics? So genetics normally is the study of hereditary characters where the character from the parent is being passed to their offspring, resembling to their parental character. And it was given by the great scientist that is called as the Mendel or Gregor John Mendel. He is the first scientist to prove the role of genetics, the role of genes in transferring character. And he experimented on the pea plant because the pea plant contains 7 contrasting characters that are visible to the eye, and when we cross it, it can be easily expressed within a short span of time by self-pollination. It is an annual plant, so that the experiment can be finished with an easy ample of time. So before going to know, we should know 3 basic terms of genetics. The terms of genetics, first we deals with about the phenotypic character. So what do you mean by phenotypic? It is the physical or the morphological character, and genotype is its genetic form, genetic character. I make you know by giving you problems. And most of the students are confused with the alleles. What do you mean by alleles? It is the alternate form of the same gene. That means if we take the height character, tall and dwarf, so these are the two genes of the that is the height character. So, or the tall character. So same genes here having alternative forms are called as what? Alleles. They are corresponding position on homologous chromosomes. So before going to this, now we should know what are the 7 basic characters given by Mendel in the pea plant that helps him to succeed in his experiment. Like the first, we should know the characters. It's a dominant trait and it's a recessive trait. So accordingly, the first one is the stem length. Here he took the stem length, dominant tall and recessive dwarf. Second, he took the position of the flower, that is axial and it is terminal. Then he took the pod shape, that is the inflated or it is constricted, swollen or it is constricted. Then he took the pod color, that is green or that is yellow. Then he took that is the seed shape, we mostly use round and wrinkle. Then the sixth one he took the seed color, that is yellow and the green. And the last one he took is the seed coat color, that is gray or white. So these are the 7 contrasting character given by Mendel in his self-pollinated experiment on this pea plant. So now we should deal with directly with go into the monohybrid cross, and after that we'll go to the problems of different variation asked in your entrance exam. So first we should know that we are having a parent of homozygous tall, having the capital T as its tall, and a parent with homozygous dwarf. When we cross both of this parent, we got the result in this Punnett square as 100% all of them are tall in the offspring, and the offspring are of homozygous, are of heterozygous format. So when we self cross the result of this, when we self cross, because it's all 100%, so when we self cross the result of the F1 generation, that is when we took the parent, one of the parent as heterozygous, that is this heterozygous tall, and again we took one of the parent heterozygous tall, and when we self cross this, and we got the result as 25% is homozygous tall, pure tall, whereas 50% is of heterozygous tall, and rest 125 and rest 25% is of homozygous dwarf. So both of this Punnett square of monohybrid, taking single, mono means taking single character. We have taken only the height of the plant, and we are hybrid. We have self crossed it, hybrid cross. And what are the result we got? All are tall, so 100% result. Here there is no probability is found, so we can't use probability in this the first table. In the second table, what happens that we got some genotypic ratio, that is the tall is out of the four square, we got one is pure tall, and heterozygous tall we got 2 out of 4, that is 1 2, 1 by 2, and dwarf 1 by 2. So when we write the format in the ratio form, 1 pure tall, 2 that is heterozygous tall, and 1 that is the recessive tall. Sorry, recessive dwarf. So this is is genotypic. The what are the genes present that control this contrasting character? To find the phenotypic character, we know how many talls are present. We got one pure tall, one plus 2 we got impure tall. Here is 2 impure tall, that is the heterozygous tall. So 2 plus 1 total we got 3 tall. So we have written 3 tall and only one we got the dwarf. So this phenotypic ratio is a morphologically when we see, we can see 3 tall plants present in it. Okay. Now next move on to our next part, that is the dihybrid cross. And a dihybrid cross, di means we took two contrasting character and self cross them to get the results. Here we have taken T, this T stands for the height and this P stands for the color. So once we have taken one character, that is the height is taken, another character is the color is taken. So both height and color are taken as the contrasting character, and this is the F1 parent, that is the 100% result will get the 100% result. That's it. This is the F1 parent 100% result, that is tall dominant and purple. We have taken the color purple and height as tall. Similarly, in F1 generation we'll get all of them tall and purple. So when we go to the F2 generation, first we should find the gametes. So how should we to find the gametes with the help of this Punnett square? Take the first one, this capital T constant. With this we got this gamete, then capital T with small p, we got this gamete, then keeping small t cross with this, we got this gamete, and then small t with small p, we got this gamete. So we got four gametes with four different color. So definitely this four gamete itself represents the ratio will come in four format. Likewise, capital P, capital T, taking one of them constant, then small t, capital P, this, and we get the this part gamete. These are the gametes. So it is a long and lengthy process. This Punnett square is the lengthy process. As you know, this white color, there are 9 white color patches present and 3 blue color. So 9 is to 3, 3 yellow color again is 3, and 1 that is pink color dwarf. So 9 is to 3 ratio, and it is a hectic part at the time of entrance. It will create problem. If any of the character is being wiped, any of the letter is being missed, then the ratio will be in different form. That will be wrong. So I will make you know the easy way to learn the dihybrid cross. How to get the dihybrid cross without using the Punnett square. So let's move the easy way to solve the dihybrid cross. First, we take an example. Suppose we take two parents, one of them having homologous parent, having tall as height character and purple as the flower color character. So in the F1 generation, we have two characters. We have taken two characters. That means they are dihybrid. In F1 generation, we got the genotype, all the genotype, 100% genotype as capital D small t, capital P small p. That is, they are the tall and purple in nature. All are tall and purple characters. So when we self cross it, when we self cross the F1 generation, that is this F1 generation, we get the offspring in the F2 generation, the ratio of this as dihybrid. So that we have already discussed in the last part, that is our dihybrid cross ratio. But how easily we can get it? Solve dihybrid with monohybrid. With the help of monohybrid, how easily we can find the dihybrid cross. For example, I'm writing the monohybrid cross that I have done earlier. So this is the monohybrid for the, I am taking the, this is a height for the height. This is the monohybrid I have taken. Next, I move on to the next purple color, same monohybrid. I will separately do the monohybrids of both these characters. So it is the color. So this one is the color, and these are the what it is the height. So I have taken two characters separately. I have found out their genotypic ratios. These are the format ratios. So what will be the phenotypic character of both of them? Normally, capital T will control tall, small t will control dwarf, capital P will control purple, and small p will control. So these are the phenotypic character of monohybrid, and this was separately. This dihybrid ratio I'm going to get it, and the dihybrid from monohybrid. So I've separated the dihybrid's monohybrid separately and found out the ratio with its genotypic and phenotypic character. Now let's move how to find the dihybrid ratio from this particular informations. So to find the dihybrid ratio without using the Punnett scale easily, that will help you in the entrance exam. How? That is, first we should write the ratio, phenotypic ratio of tall and dwarf. First height we should write about the heights, tall and dwarf ratio, that is 3:1. Then we move on to then we'll write the second monohybrid ratio, that is the purple is to white. Its monohybrid dwarf ratio is this much. After writing this is the main technique. First we should find the monohybrid. Then you can easily get dihybrid, trihybrid, tetrahybrid, everything you can get from this simple example. First you multiply tall to purple. First multiplication tall to purple, that is 3:3 will be first be multiplied. Then this purple, then we'll go for then crosswise. Then purple will be multiplied with this. Then 3 will be multiplied with 1. Got it? Then crosswise, this multiplied with this. Then 1 will be multiplied with 3. Simultaneously, this will multiply with this. And this will multiply with this. So what will get the result? Let's see. 3 will get 9. Yes. 3 1 will get 3. 1 3 will get 3. 1 1 will get 1. Easily we get the ratio without using the Punnett square. That means first we'll get tall purple, that is of 9. Yes. Tall purple 9 out of 16. Then we'll get tall white, that is tall purple. Okay. Then we can get tall white, that is 3. Tall white, yes, that is 3. Then we get purple dwarf or a dwarf purple, that is this part, 3. And we get white dwarf or dwarf white, that is this part, 1 is to 1. So easily we got, without using the Punnett square, we can find dihybrid cross simply, very simply we can get the phenotypic ratio without using the Punnett square. So friends, in our next topic, next video, I will teach you how to find the genotypic ratio and the trihybrid problems easily without using the Punnett scale. Easily with the monohybrid cross, we'll use the monohybrid application and some probability will be used. I'll make you know, and you can solve easily. Very tough problem you can solve easily, and I'll also practice the problems with you. So friends, in our next video we'll know about the genotypic ratio, how to find a genotypic ratio and some problems relating to this genotypic ratio. So all these are related. You can go to a site and get all information, all character, or what kind of MCQ, whatever you can get for needed for entrance exam, you can get from it. Thank you.